All right, so we get to learn about sour rot today. Here you go. So sour rot of peach, is it an emerging issue or is it a one-time kind of thing? And that's a question that hasn't been answered yet, but I'll give you an update of what actually happened over the last few years with regard to sour rot of peach. So peaches obviously can be attacked by a lot of different fungi, and they can cause actually similar symptoms, especially early in the symptom development. Now on the left-hand side over here, you've got, like, for example, brown rot, and that's an advanced lesion. You can basically tell that this is brown rot based on the spore symptomology. Got anthracnose, rhizopus, more Colotochicum species over here. But in the early stages of symptom development, they all kind of look similar, and it looks like a brownish discoloration that is expanding in either roundish shape or oval shape on the peach. And this is basically what one farm in South Carolina has seen over several years now, being a problem at their location, at their packing house. And we were trying to figure out what the problem actually is. It turns out that the problem wasn't cold damage, or wasn't brown rot, or wasn't Colotochicum. It was sour rot of peach, something that we don't really see that often, but it's seen quite often in California. And if you go to an orchard, for example, that is neglected, or where the grower chose to not harvest for one reason or another, you would see peaches like this that decay on the orchard floor, forming this white symptomology, and oftentimes that is caused by sour rot, and the causal organism is Geotrichum candidum. On culture or in culture, it looks like this. It forms a white mycelial mass. Under the microscope, it looks like this. It produces these hyphal tips and hyphal structures, and those are the spores that the fungus is producing. It's not really a true fungus, it's a yeast-like fungus. So it's not a true yeast either, it's a yeast-like fungus. So in order for us to understand a little bit about this disease, we need to learn a little bit about its behavior in the field, its survival, its dispersal, infection, and reproduction. So let's look at the survival. Survival can happen through the mycelium and the spores. In the field, the fungus survives on plant debris in the soil, on leaf and tree surfaces, and in the packing house, it survives on soil and plant debris particles, on infected fruit, and on bins, and in the packing house, and on any packing house surface, basically. With regard to dispersal in the field, it is mainly dispersed by rain splash, so it's not really picked up by wind. It's just a very short movement from one area to another through rain splash, but it can also be moved by insects, specifically sap beetles, for example, sometimes even fruit flies, if there are fruit flies in the orchard. It is a pathogen that does require wounds, and we tried infecting actually peaches without wounding, and that is very hard to do. It does require wounding, and wounding can happen through abrasion while handling the fruit during harvest, transport, and storage, and also because of insect damage. And one other interesting fact is that the fungus only affects maturing fruit or mature fruit. You don't really see it ever on green fruit. With regard to reproduction, there's no sexual stage that I know of. There's just the asexual cloning of the fungus, so to say, and it sort of clips off little pieces of its mycelium, like you can see over here. That's the hyphal system, and then it just snips off some of those strands and forms these arthrospores. Now, one interesting fact right here that is actually was put together by my co-author Julia Gillane. This is a fungus And here's where you wake up, Greg. This is a fungus that you actually have in your guts. It's one of the thousands of gut microbes that do whatever they're doing in the guts. And it's also something that you might have eaten already if you do like camembert cheese. Camembert and other similar cheeses are fermented with this fungus. How do we usually control sour rot? Well, in the packing house, we typically have two options that we can use. We can use Scholar and we can use Chairman. And those are the two chemical options. There are a few more non-chemical options, but those are the most widely used chemical options. Scholar just consists of fludeoxonil right here. And it works for brown rot, rhizopus, anthracnose, but it does not work for sour rot. Fludeoxonil is not effective against sour rot. On the other hand, fludeoxonil and propiconazole, which is sold as Chairman, controls brown rot, rhizopus, anthracnose, and sour rot because of the propiconazole component. Propiconazole is what we call DMI fungicides, demethylation inhibitor fungicide. So several years in a row, this particular packing house has reported problems with sour rot outbreaks. And they started as these small lesions that I already showed you, and then expanding into something like this. If you leave the peaches for a week in the carton, this is what happens. They're being totally consumed by Geotrichum candidum. It smells sour, and it produces the white mycelium on top of the peach. And that is all these signs and symptoms are consistent with sour rot. So for us, in order to do a proper study, we had to collect isolates from the packing house, which we did over several years. We also collected isolates from around the packing house. We call those isolates South Carolina packing house if they were from the packing house, and then South Carolina non-packing house if they were collected from around the packing house. And we also asked some collaborators of ours to send us some California reference isolates, which it's always good to have if you do a study like this. And so we took those isolates and we grew them on artificial medium that was amended with different concentrations of propiconazole. And then we measured the expansion of the colonies of these isolates, and that would give us an idea of how sensitive they are to propiconazole. And those are the results. Let's focus first on the sensitives. So we basically saw three different phenotypes: sensitive, reduced sensitive, and a resistant phenotype to propiconazole. I'm always talking about propiconazole with this regard. So the sensitive isolates, they were from California, and they had a very low EC50 value. EC50 value is basically the dose rate that is required to inhibit 50% of the mycelium in a petri dish. That's called the EC50 value. And the MIC value would be the minimum dose required to completely inhibit mycelial growth in a petri dish. So we found that the California isolates were consisting of two groups, one that were super sensitive, very low EC50 values. Then there were also some reduced sensitive isolates in the California selection, and they had slightly higher EC50 values, about 10 times higher. Then we found some reduced sensitive isolates in the South Carolina non-packing house bulk, and we also found resistant isolates, and they were all from the packing house. So there was a clear difference in vitro results among these three different groups of isolates. And again, we also tested fludeoxonil, and fludeoxonil had absolutely no effect against those. All right, so we found out that there are differences in sensitivity to propiconazole in those isolates from the packing house. So what does it actually mean? And for us to study this a little bit more, we need to dive a little deeper into modes of action of DMI fungicides. Now, this is a picture that just illustrates that there are different chemicals we're using in crop production. You know, this, for example, is thiophanate methyl, and then there's SDHI fungicides and other fungicides, and they all have their very specific mode of action. Now, the DMI fungicide, which includes propiconazole, that one inhibits a specific enzyme in the fungal cell called the 14-alpha demethylase, short SUB51. And if that one is inhibited, it messes with the structure of the cell membrane so that the fungus really can't expand anymore and dies. There's one target, so one of these enzymes is being targeted by the DMI fungicides, and that enzyme is called SUB51 or 14-alpha demethylase. There are several types of these SUB51 enzymes. There's an A version, B version, sometimes there's a C version. Now in our case, we're interested in the B version, so we add this B letter at the very end of this gene or this enzyme name. And if you're talking about a specific fungus, like for example Monalinia fructicola or Geotrichum candidum, you add the initials of that fungus to the gene name, and we end up with GC, Geotrichum candidum SUB51B. So that's the enzyme that we were really interested in looking with regard to this study. And so we were able to actually identify this enzyme in those isolates from the packing house and the isolates we collected around the packing house and the isolates from California. And we found some interesting differences. So between the sensitive isolates that were highly sensitive to propiconazole and the reduced sensitive isolates, we did find one mutation in that gene. That's the illustration of a gene, very simplified, consists of a whole bunch of amino acids. And the only difference between the sensitive and the reduced sensitive isolates was this change in amino acid from Y to F, actually from F to Y. Then we looked at the structure. So we found those genes in California isolates over here and in the South Carolina non-packing house isolates. The isolates that came from the packing house, they were even more unique. They contained two more mutations, that one, and all three together may be responsible for this unique phenotype, for this unique behavior of these fungal strains, and they are completely resistant to propiconazole. So there's some genetic basis to it. So then the question was, okay, does that really matter? Is it relevant that they have these mutations? So we did a little detached fruit study. This was done on cherries because we had cherries available. And over here on the left, you can see a representative of a sensitive isolate right here. And that's a reduced sensitive isolate, and that's a resistant isolate over here. And if you don't treat the cherries with anything, they all are pathogenic and cause sour rot of cherry. But if you treat these cherries with propiconazole at the rate that we are using in the packing house, you can clearly see that the sensitive isolate is completely controlled, which is what you like to see. On the other hand, the reduced sensitive isolate produces a little bit of a lesion, and the resistant isolate didn't care at all about this chemical application of propiconazole. So it's completely resistant. So it matters. So then the big question was, okay, where does this resistance coming from? Was this resistance selected in the packing house due to frequent application of chairman, for example, and selecting for resistance in the packing house, in which case we wouldn't expect resistance to see much out in the field, at least not where none of the cull fruit was deposited. On the other hand, It could also be that over decades of spraying DMI fungicides such as Indar and Tilt and Orbit and these kinds of things, that resistance was selected in the orchard, in which case you would expect to find resistance everywhere. So we looked at this and went to several different locations, several different orchards. Those are all listed over here. Half of them received no cull fruit from the packing house, and the other half received cull fruit from the packing house, which basically means that the fruit that couldn't be sold was moved back into the orchard. That's what I mean by cull fruit deposition in the orchard. And so we looked at 37 isolates that came from no cull fruit areas, and then 34 isolates that came from areas and plantings that received cull fruit. And here are the results. Basically, the distribution of these phenotypes and genotypes was the same. So in the non-cull fruit and in the cull fruit orchard, we found resistant isolates, reduced sensitive isolates, and sensitive isolates. Looks pretty much the same, which indicates to us that the resistance probably was slowly generated over decades in the orchard, and that it's probably widespread in orchards of that particular farm. And it also means that sterilizing your packing house and trying to get rid of those resistant isolates is not going to help you much. It may reduce the incidence of maybe sour rot, but it's not going to eradicate the problem, because the isolates are in the field and they're continuously brought in with the new peaches that are being brought into the packing house. So then the next question was, well, I mean, propiconazole is a pretty old DMI. That's one of the first ones that we had. It's been registered since the 1990s, 80s? I'm not sure how old it is, but it's one of the first ones. So it's been around for a long time. And since then, we have gotten many more better DMI fungicides with generally increased intrinsic activity against the target pathogen, which is brown rot of peach. So we looked at the efficacy of tebuconazole, mefentrifluconazole, fenbuconazole, and difenoconazole, and you probably recognize these trade names: Elite, Sevia, Indar, and Inspire. And we were trying to figure out if they work better than propiconazole. We also added a few other treatments, including quaternary ammonium, like agwash, polyoxin D, which is an extract of a bacterium that is also registered in some fruits for post-harvest treatment, netamycin, which is a common product that is being registered for disease management in fruits. And we also looked at some Pseudomonas and Bacillus strains that are commercially available. All right, and here are basically the results. So I'm going to focus first on propiconazole right here with this color, like beige. The sensitive isolate compared to the sensitive isolate was producing no lesion when treated with propiconazole, which is to be expected. The reduced sensitive isolate produced also basically no lesion when treated with propiconazole, but the resistant isolate did produce large lesions that were pretty much consistent with the untreated control. Now let's look at the other DMIs. So that's the response of the other DMIs, and you can clearly see that the bars are much higher compared to propiconazole. That means they had no effect on the growth of the fungus. Not here, not here, and not here. So the sensitive, the reduced sensitive, and the resistant isolates were all not reacting to those DMIs. So that was kind of surprising for me that not even the sensitive isolate, which is sensitive to propiconazole, wouldn't react to those DMIs. But I guess there is a reason why they chose propiconazole for post-harvest treatment. It's just the one DMI that works against yeasts. We also looked at polyoxin D, natamycin, quaternary ammonium, Pseudomonas, and Bacillus, and as you can see, there was just not much effect here. It was not significantly different from the untreated control, not the sensitive one, not the reduced sensitive one, and not the resistant isolate. So nothing really works against sour rot other than propiconazole, unless you've got resistant isolates. So in summary and in conclusion, Geotrichum candidum resistant to propiconazole is widespread in the soil of this specific South Carolina farm that we studied. Chairman, which is a combination of fludioxonil and propiconazole, is not effective against these mutated resistant strains that we are recovering from the orchard soil, which opens up a problem, right? So that could in the future be a problem. Unfortunately, nothing we tested so far, conventional or biological, was effective against sour rot. So we don't really have an alternative to propiconazole right now, but we keep looking. And the resistance is stable. The resistance is based on a point mutation in that enzyme that that we're targeting with the DMIs, and therefore it's here to stay. And in conclusion, where resistance is a problem, we've got to focus on sanitation. We've got to focus on sanitation of bins and storage facilities. We've got to make sure that we prevent injuries of the fruits before and after handling, and we've got to control insects as much as we can. And we certainly, again, need to find alternatives to propiconazole when it comes to sour rot management. And with that, I want to thank all the people that were involved in this research, Dr. Julia Gillane, Bing-Yu Chow, Sarah Price, and Tonya Blanc, and thanks Sarah Oswald-Scott for helping us find all those orchards. And with that, I'm opening it up for questions. No SDHIs are working against yeasts, no QIs are working against yeasts. Well, Captain is a broad spectrum chemical. Yes, it will have some effect, you know, but Captain is also an anti-sporulent. Yeah, against the spores it would have an effect, I would think, yes. But Captain is not something we would use post-harvest, right? No, yeah. Yeah. I was hoping I would find a better DMI fungicide that we might be able to use pre-harvest other than propiconazole. We could focus on that one, and just there's nothing there. Yes? Well, so we tried that, right? That was in the last few slides. We tried non-chemical solutions like biologicals, like Bacillus, biological extracts like Pseudomonas. Physical like what? Oh. Like UV light and stuff like this, UVC. Well, yes, UVC light is non-selective, and that would work. Yes. Yeah, UVC light would work in the packing house. Well, thank you very much for your attention.
All right, and next, Dr. Ksenia Gasek is going to give us a little update on her breeding program and some new releases.
Okay, thank you for coming to this afternoon session of the last day, and thanks for inviting me to talk. This talk is going to be more or less similar to what I told you last year, because it's just 1 year, so there's not much to do within a year if you're a breeder, but I try to reshuffle the way the information is presented to help you more with understanding the differences and provide you some ideas of what might be coming in the pipeline. So basically that's going to be the outline. I'll just shortly revisit the breeding goals just so you understand what the program is aiming for, and then cover the newly patenting cultivars, and then talk about what's in the pipeline and what we learned about them in a few years that we included them in replicated trials. So basically, South Carolina, as you know, is the second in the nation in production for fresh market peaches, and that's why the industry wanted a breeding program to address the issues that they have in the southeast U.S. And so my program was established in 2008 to merge the high quality of peach cultivars with the disease tolerance, because anything that's bred in other parts of the country and the world, when it comes to our region, usually fails due to the humid conditions and the pest and pathogen presence that we have. What we are trying to address through the program is to actually address all of these challenges that you're dealing on a daily basis through various ways, and some of these are easier to achieve, some of these are harder. And one that we still did not move much is the retailer education, and I'll address that and connect it to the Dr. Iglesias talk this morning regarding the harvest and types of peaches that are being released. So this is the ideal peach I showed you several times already that we are trying to achieve depending on the ripening time, because what you asked me to do is to try to cover the ripening period as much as I could and provide for each week some options for you to compare it with what you grow right now and to see if it's better or if you can maybe diversify that way. And so addressing, of course, the size in the early material and no splitting. Then throughout the season is the tolerance to the diseases. And in the previous slide, I didn't emphasize that the two diseases that we focused at the beginning were bacterial spot and brown rot. So those would be, when I say disease tolerant, those would be the ones that I'm actually referring to. So just to remind you how long it takes to breed the peach or any fruit tree cultivar. And so peach is on the better side of the fruit trees when it comes to breeding, but still takes a long time. And so right now my program is in past 15 years, we are in the stage when we actually send material to the replicated trials. So we give you, as growers, the chance to see them and to give us the input, what you like, what you don't like, and what you want to change. Here are some numbers through the years, including the 2024, of how many crosses were made, how many seedlings generated, planted, looked into the field, and the number of 650 actually now is like maybe 350, because when I started my program, we did not have conditions at the Master Farm to see the bacterial spot. Now we do. And so that's why anything that I saw in these early years that I liked for the quality and taste and everything, when it failed miserably for the bacterial spot, it was out of the planting. Also, for some of the, if you remember previous presentations in various conferences and meetings, there were much more advanced selections, including in replicated trials, that actually showed susceptibility, so they were removed as well. Since the quality is really good, I'm using them as a material in breeding to improve, but they are no longer mentioned in these type of presentations. So I mentioned the replicated trials, and these are the places where we planted material. Arkansas was included just at the beginning for few of them because they really have a high incidence of bacterial spot. And at the beginning, as I said, I didn't have it in Master, so I wanted to make sure that my material will be exposed to the bacterial spot, just to see if what I'm seeing or trying to get, I'm reaching that. In Alabama, the results you could see in a poster that was outside. Dr. Elina Koneva prepared the poster with the new tree releases and how the performance. And what I'm going to be focusing today is mostly data collected from South Carolina plantings. And there is one that because it's a picture, I couldn't delete this one. I was trying to establish the orchard in Sand Hill Rec, but it didn't work, so that one doesn't exist. And the newest planting is in North Carolina that was harvested first time last year, and unfortunately revealed some bacterial spot susceptibility in material that we didn't see in Alabama and in South Carolina. So that you get with the replicated trials, because you never know if the conditions are the same or isolates of the pathogen are different and they might affect it differently. So when I say that something is tolerant, I will point out where those were planted, just so you know where we actually tested them, and where they actually stayed tolerant through the testing phases. So now we're going into the new releases. So as you remember, the 16 years, we are finally in the phase that we actually release something. But before I list them, I want to explain the naming behind these new releases. It was a tough battle to find how would I name the new cultivars. And I wanted to keep the tradition of the Clemson material released before me, but still had a problem because Carolina is used in North Carolina, Tiger is used in Texas, so you had to kind of find a niche, and the Caro used to be used in Clemson for Caro Red, Caro King, and etc. So I wanted to keep that and merge it with RES, R-E-S for resistance, and some descriptive word about the accession. And Caro means that those are the regular type of peaches, fast softening what you expect, what you used to. The Everest is mentioning the type of peaches that hang long on the tree, and those would be the ones to help you with the 2D orchards and automatic robotic harvesting to harvest in one go, because you can let the fruit sit on a tree for a longer time without losing the quality. Actually, it gains the quality, and wait after 2 or 3 weeks from when you would normally harvest to harvest it at one go, and you would not have any culled peaches because you waited that long. Those are the special ones that are firm like a rock. They stay on a tree for a long, and the moment they picked from the tree and put on the kitchen counter in the room temperature, they will require sometimes half a day or 2 days to actually soften and then be melting, juicy, freestone, anything you would expect from the peach. But if you pick it from the tree and bite into it, it's like an apple. And so there are people that like that, there are people that don't. So just to know for those, and there are many in the market, but you're never told. Unless you go and read the fine print in the patenting documentation, you would not know that. So these are the 2 selections that are already patented. The Carores Wonder used to be tested as SC2. In replicated trials, a patent was awarded in July, and the Everest Ruby was tested as SC1. Patent was awarded in August last year. Those 2 are the examples of what I just described. Everest being the standard type of peach, fast softening, and opposite Carores, and Everest Ruby being the one that requires few days at a room temperature to soften before you would eat it. So when I went back to the retailer where we still failed to educate them, we know growers produce excellent quality of peach, but we also know we don't get that in the store. And so there is a miscommunication somewhere and mishandling of the fruit, and especially when the fruit is of a different type, fast softening or slow softening, that we need collectively try to make an effort to educate the retailers how to handle that fruit, so the growers can translate the great quality to the consumers, and consumers come back and buy the fruit. And the third one that's now in process of patenting is Carores Delight. So it's the same type, fast softening peach melting that we resubmitted the paper, so I'm expecting any day now to get actually approval. So those are the 3. All of them are yellow flesh peaches. You see high blush coverage, and those are early ones with earliest one being Carolus Wonder, ripening like around first half of May. Then Everest Ruby around first week of June, and then Carolus Delight. So this graph shows you days before or after, later on, July prints, because I think that's more relevant to you than if I give you the dates, because Master Farm is kind of in the upstate, so it's not really close to the main growing region, so it's like a 1 week or 2 weeks difference. So what's unique about these is that they, if you remember in the ideal peach list, the goal for my breeding program is to keep the chilling requirement from 600 to 800 chill hours to be sure that they're going to get their chill satisfied. So you can tell that I kind of miss the goal, hit the goal, and miss the goal. And so that's kind of the thing that you experience. But chilling requirement is not the only thing that's important. We want to also control when they bloom, and we want to push the bloom to be after the in the first week or after the second week of March, which is what this line indicates. And here is the like a range from 600 to 800. And unfortunately now I think it actually we need to go even lower, maybe 550 to 750. So what I want you to remember here is where the Carolus Wonder is. So it has the 500 chill hours requirement, and it blooms earlier. So this is Julian days, so 60 days is like 1st or 2nd of March, depending on the year. If it's a leap year, then it's a 2nd. And so it blooms sometimes almost in the February. But when we look into the production, which would be, okay, this will actually tell, but replicated trials, we were freezing for, let me go back so it's more evident. We had loss of crops for Everest Ruby and loss of crops for Carolus Delight, but not for Carolus Wonder in the same given year. So we knew there's some difference because it's the lowest chill, earliest bloom, you know? So how come it produces when these that are medium or higher chill and bloom later, they get frozen? Dr. Melgar and I were looking into the fruitlet freeze tolerance, and in this graph you see them ordered one next to each other. And if you look at this dotted line, it shows you the middle point when half of the material we tested was damaged. And that's kind of the standard way you report on how they tolerate the freezing. But that actually doesn't tell you the story, because if you look at this line, how there's no damage at the beginning and then the damage starts at -4. Keep in mind this is in the control conditions, so probably in the field that's going to start earlier. But look at this. Almost immediately as the temperature goes below 0 degrees Celsius or below 32, the SC1 or Everest Ruby starts seeing the damage on the fruits. And the Carolus is doing much better, and Carolus Wonder and Carolus Delight. So that's what I was showing here with this graph. So even though the combination of chilling and bloom would indicate that they should or should not be more tolerant to the late frosts, the problem is that the late frosts are coming at the same time. What's not the same is the stage where the fruit is. So if the fruit is more developed, it's going to get killed by the late frost. So if the fruit is more tolerant, you have a better chance to actually have the crop. So that's where I'm going with these. And here is just a slide to show you how they look in comparison with the closest standard that you are growing. And on the top is the Julian day for ripening, or the date again at the Master Farm. And this, like how the fruit looks. And you can tell, I try to make them so because each of these squares is 1 inch, to have the same size, so they're comparable, so it's not like a bigger picture or smaller picture. And you can see how Carolus Delight, or SC9, if that is what resonates better with you, for if you tested it, it has a great size. So I am showing you the performance versus standards, but just for Seneca, because last year, due to some issues in my program, we were not able to collect the data from all replicated trials, so I just decided to stick with the Masser farm where I actually collected the data. And so you can tell that they are either similar or better in the performance than the standards at the Masser farm. But what's more eye-opening, and thanks to the Titan farm and the packing house data, this is actually what we wanted. So I don't know how well you see there. From my angle, it's not really well seen. It's better on the screen of the computer. The colors are not very good, so hopefully the recording will show if you rewatch it. But what I wanted to for you to pay attention for the Carol's Delight, so these are the categories of the fruit size, and Carol's Delight has 100% fruit in the 2.5 to 3 inches size. It does not have a smaller or higher size fruit. And the similar is in Everest Ruby. Everest Ruby has a 3 1/4, which is like the tiny region over here, but does not have fruit smaller than 2 1/2. And from others, that was just the summer prints that had that category, and some portion in the Ruby prints. So it's really great pack out for them, which is rewarding, considering when it's what time of ripening it is, when we are suffering with the smaller size. So these are the 3, and they're licensed to Cumberland Valley Nursery and Vaughan Nursery. So if you have interest in them, you can contact them to get the trees. Okay, so now that we covered those 3 that we released, and I just want to shortly, before I go into the future possibilities, thank you all for helping me coming to those 3 to release, and that's basically the growers, the industry, everybody that looked at them and tested them. So, future possibilities, that's really tough choice for me, because there are so many. It's like picking your children, so which one you want to showcase. And so now what I try to do, since before this talk, most of the material was actually early ripening. And it seems that the early ripening material is, at least for me for now, was more easy to get tolerant to bacterial spot than the later ripening. And of course, I did not emphasize, but all 3 of those are tolerant to, that I just showed you, are tolerant to bacterial spot in fruit. SC1 was tested in Arkansas, Alabama, South Carolina, North Carolina, was clean in all places, or Everest Ruby. Carol's Wonder was tested in all of these places, and Guido was able to find 1 fruit with 2 dots on the bacterial spot. So I never saw it really hammered, except last year in Arkansas. But not in North Carolina, not in South Carolina, not in Alabama. So we think, and with Dr. Hehe, we are testing those isolates to see if that's due to the isolate, just so I'm not misleading you when I say that it's a tolerant. And SC9, so far, we did not see any damage anywhere. But, I mean, there's time to find it. And so on this graph, you see anything that has SC and number is something that's already included in replicated trial. Anything that has a full name, because this is like a Dr. Oki's labeling system, where it's 2-digit year, 2-digit cross, and 3-digit individual 3 number within the cross. And so when it has that, those are my choices to present to the industry and see if they like what they see and if they want to put it in the replicated trial and look how they look. So I focused this time in something that's later than July prints, because that was basically what was missing from previous presentations. So I will not go into the page by page for each of these. What I'm prepared for you is comparison between them, just so you see where the fruit quality is, or size, like in this slide, this is the diameter. So the dotted line is the 2.5 inch, and the solid line is the 3 inch. And so these are all of those, and here is July print, so these are these new, just so how you can see this. So of course the early stuff is around average size 2.5 inches, but the later stuff is between closer to 3 and sometimes even over 3 inches. And I was told by the growers that they don't actually want it higher than 3 inches. With now COVID issues and everything, with the different way people buy fruit and packaging and everything, the 3-inch size is what is preferable, not huge ones, because we do have a huge ones, but you don't want to sell one peach, you want 5 or 6 in a package. So here is the fruit weight, and of course that's comparable, and you can see that later ones across around 200 grams. And so all of them are yellow flesh, all of them are peaches, and all of them, except the like SC1, 9, and 2 are released. SC1 and 8 are the slow softening type. The rest of them are the fast softening type of peaches. And chilling is around 800, so we were able to keep that for all of them. And also, all of those have tolerance to bacterial spot at a master farm again, especially these, since I had no chance of testing them. Among these, okay, I'll just show you something about the bricks, because that's important, about the sugar accumulation. And you can see from the July prints and afterwards, they go over 12, 13, and even 14 bricks on average. And I can tell you, you do better job than we do at the master farm, definitely when we harvest and how we process. So the numbers that I get from replicated trials are always better performing than in my trials. And this line shows the pH, so they are all standard type. That's what I forgot to mention. They're all normal balance between the sugars and acidity. There are some that are with a lower acidity. SC1 is on the lower side, but not really low, so it's not going to be, but there are some in the pipeline that are with really low acidity, so you have a perception that the sugars are high, but when you measure the bricks, it doesn't actually reflect that. It's just your taste buds feel that there's not that much acidity in the flesh. And so now I'm going to show you a few, okay, so why is that empty? Okay, so how few of those past the July prints look, and you'll notice one trend, that there is a lack of coloration, which we are trying to kind of address. They still look okay, and they still have just a lighter blush than normal, and then in the corner you see the days past the July print, so you know, and here is, I managed to find the actual picture of the fruit on the tree. And then this is the other one. This one's really, in this picture, not really reflects how nice the fruit looks. And this one is the one that we are testing as a processing peach, because the Titan Farm was asking us if we have anything that they could test for their processing facility, and then we and the main requirement was the high sugar accumulation, and this is the one that had 14 on the graph, and that's why we selected it, because we didn't like it for a few things, but now it seems to be performing better when you take better care of the trees. That's kind of the trade-in when you're in the breeding program. And so some of these on the previous slide are also shown to you few years back when I started showing this slide in our brown rot test. So brown rot is a harder pathogen to deal with than the bacterial spot as from the breeding perspective. And we were able to get some material that's more tolerant, but not resistant. And I don't think we'll ever have that. But what now Dr. Schnabel and I will try to do, we have an orchard, which the fruit came from that orchard, and it's shown here. And the first one is Dr. Chen's Rich Joy cultivar, is still under its code name that we included, and that's the orchard that never received any fungicide. It was just insecticide, and those are now 70-year trees, and of course they suffer from monilinia or brown rot, but you can still see that there are some that this shows you the 3, 5, and 7 days post-harvest when they clean fruit put in the humid area just to see if the monilinia will develop. And so you can see that after 3 days, 5 and 7, it's still much, much less than these or the control. And here is China Pearl, and here is O. Henry, Red Globe. And you can see how the fruit looks. The origin of the tolerance is from one of Dr. Oki's selection that is very low blush, the clingstone small, and so that's hard to take away. This particular one is also tolerant to, or actually resistant to bacterial spot. So which is nice coincidence that we got into the same material, both tolerances, so we can use it as parents. And then this one is the one that looks the best as far as the visual performance and the taste. Unfortunately, it's not bacterial spot tolerant. So we are using it in the breeding to improve, but this one has a really nice performance for, okay, this moved, sorry. Yeah, it should have been here, but yeah. Nice performance for brown rot. So we will see with this brown rot type of selections, so I'm not promising no spraying, but what I'm saying, if Dr. Schnabel and I can show that you can maybe start spraying week before the harvest, not 4 or 5 weeks before harvest, then that would reduce your input cost, deal with the issues with the environmental pollutions and stuff, and save all of us time and money. So that's kind of the goal that goes with these. So with that, thank you all for the attention. Thank you all for the funding, especially the South Carolina Department of Agriculture, Peach Council, individual farms, and Clemson experimental station. These are, of course, I'm one person in front of you, but there is an army of people coming through that collected all of this data and worked in collaboration with me and with you to get the data, and I'm open for questions. There are 2 minutes, so.
Oh, you're fine. Yeah.
So if there's a question and you know my email, you can always find me and ask any questions. What I was hoping, if we have the time, since there was no last talk, maybe we can have also like 15 minutes or so general discussion where you would have a chance to maybe say what you want from us. I'm not the only breeder in the audience to look for something specific or something, but we'll have a chances in these upcoming meetings where I'll more rely on your input than today. So thank you.
And Dr. Phil Brennan's going to talk to us about anthracnose and brown rot. Is yours on here?
All right. Good afternoon, I should say. So I'm going to be covering some research from the last year that we have been conducting and looking at a couple of problems, mainly the peach anthracnose and also brown rot, and adding in especially some of the new chemistries that are involved in brown rot management in the future. And first I'm going to talk to you a little bit about peach anthracnose. And so this is really a complex, I guess is what you'd say. It's Calototricum acutatum and Calototricum gliosporoides complexes. So there's a lot of different fungal species that are probably involved in this if we really look at it. As far as the symptomatology, initially the lesions are very small, brown spots on ripe fruit, but they become darker, circular, and slightly sunken. As you can see here, that's what they look like. And as these lesions advance, they're actually very firm, which is not a traditional kind of a rot, but they are brown, sunken, and then you have these concentric rings, which you can see here with these salmon-colored spore masses. And here you can just see an example of the types of spores you can get out of there. As far as the disease cycle, we think it overwinters mainly, I think, on bark crevices and probably old cankers and things like that that are in the tree itself, but also possibly on old mummified fruit on the ground or in the tree. And then spread is generally by rain. Warm, moist weather around 77 to 86 degrees Fahrenheit will be optimum for development, and you don't have any real development of this disease when it's very cold, below 40 degrees Fahrenheit. As far as management, things that we can do, make sure you don't have a whole lot of legumes in your actual orchard, as that's an alternative host for the pathogen, and then also wipe out any wild printed species that are close to the orchard as well. There does seem to be some variation in susceptibility among varieties, but I don't know that we've ever really rated that. But there is a difference sometimes among the different cultivars as far as susceptibility. And then the materials that are actually active right now that you can look at that would work would be things like chlorothalonil, which is in Bravo, stuff that we would apply early in the season, and then Captan and fungicides in the frac control group 11, which are things like Abound or components of Pristine or Maravon. And at least 6 applications prior to harvest is recommended or has been recommended for blocks and varieties with severe disease. And we do see a lot more of this over the last few years. There's some work done at Clemson with Eldon there when he was there, and he was just looking at latent infections. And reading back over this old paper, he basically determined that yes, there are latent infections where you really don't see the disease until the very end of the season. But he did, with the paper itself, he could tell you that at pit hardening and afterwards, you would have latent infections that would lead to disease. But he did say he thought it would be a season-long event, that we could have earlier latent infections as well. But again, the fruit symptoms don't really show up until the very tail end of production, I mean right at harvest. So you can have what appears to be a perfectly good, healthy peach, and then you will see it start to have these symptoms. So this last year, this was really the second year we tried to do this. All our peaches got wiped out the first year we were trying to attempt to do this with the cold damage. But last year we actually had some successful on-farm trials, had a couple of farmer cooperators who helped us with these trials. And I'll try to explain these to you in some detail. But the objective was, first of all, to look at how effective is or the impact of Captan on this disease. How good is it? What can we really get out of just Captan? And then also we wanted to, again, revisit to some degree the timing of these infections, early, late, or season long, and try to get a feel for that. So this is one of the farmer trials that we had. I know this is a bit complicated, but I want to key in on a few things. Now, this was not, from the standpoint of what we wanted to achieve, this was not exactly the perfect trial, but it actually did help us to learn something, I think, as well. So if you look at this first application treatment regimen, we're calling that the Captan full season. You can see there's a lot of materials that are applied, a lot of Captan that's applied, and then you have Maravon, Lunaflex, Pristine, and Fontellus, quite a few fungicides. The second one, which is what we were calling our early season applications, the only difference here is there's really one extra application of Captan, so key in on that. We missed this one application of Captan here and went back to sulfur, which would not have any activity. And then in this case, what we called a late season, we just had all the sulfur early, and then we had just that one application of Captan. So not a whole lot of distinction between those 3 regimens, so you might kind of expect them to be the same. If you look at the rainfall, and I'll show this again later, but you had periods of intensive amounts of rainfall, and then you also had like really dry periods. And the area of time right before we were harvesting this fruit was excessively dry. I mean, there was just no moisture whatsoever. So if we were getting infection during that time frame, that would be very unusual. And as far as the site, there's some things we learned about this one site. First of all, we were able to find out last year that all of the Calototrichobliosporides, which is what was identified there, that particular complex, was 100% resistant in the QOI type fungicide. So things like Abound would have no activity whatsoever. And this was associated with this G143A mutation, which is well known to cause fungicide resistance. we went in on the 25th of June at this site, and again, we're talking about up in this area here where we've had all this dry period right before this, and we did not see hardly any anthracnose at all. And this was just before harvest. We've been called by the producer and told you have to go in and get your fruit now because we're going to harvest. And so we didn't see enough anthracnose to actually harvest anything at that point. We could find a few fruits here and there together, but not anything we could actually get any data out of. So what we did was we actually harvested fruit and put it in boxes, and it was all good fruit. There was no symptoms at all of anthracnose, and so we did this similar to what we would do with a post-harvest brown rot type trial. And then we went back because anthracnose continued to develop in the field then, and we did a tree rating on the 10th of July because the producer said, oh, we've got a lot of anthracnose. And I was there last week, I didn't see any. And so we were able to go back and actually get some additional data. But this is just observations of what we saw at this one site. And this is what I want to key in on. If you look at kind of these gray bars here, this is what we got in the box. So say what we took out of the field, took out the fruit, put it in a box, and just let it sit. And this is our full Captan. Remember, we only had like really one additional Captan application, and that's what we're calling full Captan, versus early Captan versus late Captan, which are very much the same. And if you look at tree incidence, when we went back, what percentage of the trees had anthracnose in the tree, all of these were relatively high, but here's your full Captan, early Captan, and late Captan. Again, remembering that's really only about one Captan application difference. And then when we look at the average count of fruit in a tree that had anthracnose at that time in the tree, here's your full Captan, early Captan, a little bit higher, but about the same, and then late Captan. So what I think this is telling us, again, this is an on-farm trial and you get out of it what you can, but I think it's saying to us that basically in that situation, that one Captan application, which would have occurred during a time frame which we had a good bit of rain, was a critical application for that particular trial. So it's not just a matter of whether it was early or late, it's also the timing of that particular one application that changed things. Okay, so that was one trial. This second trial on the Valley Sweet, this is kind of what we had hoped to achieve. You can see there's a difference here that's more clear. We've got what we're calling our full season Captan application. Here you got all those Captan applications, and then we switch over to Bound, Maravon, and Pristine. Here's our early season Captan applications, then kind of a clear demarcation, and here are our late season. Again, you'd had the few sulfurs in between there, but there's differences here. You can see we actually have a full season, an early season, late season. The idea being that's going to help us to determine when is this anthracnose coming in. That's why we did that. And then there's your rainfall patterns again, just for your edification. Now on this site, when we went in on 25 June, we absolutely saw no anthracnose whatsoever. I couldn't find any in the field. I could not see it. And of course, as a pathologist, that's disappointing when you put in a field trial. But we did what we did before. We took boxes, filled them with good fruit, and took them back, and then determined what would happen. And out of this, we got some really clear data. Now, given the incidence of disease is not that high, but if you lost 10% of your fruit, that's a lot of fruit to anthracnose, especially when you took in what you thought were good fruit. So in looking at this, here's our full Captan, all those Captan applications, a full season. We did not have disease. It simply was not there. We never found a single fruit that we had any indication having anthracnose when you applied that much Captan. If you applied just the early Captan, you can see that we got a difference, right? There's some disease that is coming in later, but those early sprays are going to make a difference. And then if we had put out only the late Captan, this is where we had the most disease, again indicating those early season sprays, very early, much prior to pit hardening, were actually having a major impact. So what this indicates is, yes, you're going to need some type of season-long control in order to control anthracnose in the field. Those latent infections are occurring both early season and late season, everything in between. With that said, I think there could be an opportunity to time anthracnose applications of something like Captan. This is done now in strawberries. It's done in blueberries. We've never looked at it in peaches. I'd kind of like to gauge the interest in producers. I just don't know if this is something you can use. I mean, a lot of the producers that we have really large acreages, and for them to time an application specific to 1 or 2 orchards may not be practical. But I think that would be something we could do. I don't think you're having infections all the time throughout the season. I just think you can have infections at any time during the season. If we could time those applications, that would be helpful. So what I would conclude is that full season applications of Captan where you have an anthracnose problem will work. You can definitely reduce your level of anthracnose by putting out enough Captan. If you put your finger on the trigger and keep pulling it, it will work. And then early applications can be just as important for these latent applications as the late season applications. So we have to be applying basically season long unless we can time it. And I think there is an opportunity for timing. The other thing is the QOI fungicides are starting to fail. This one site in this trial was not the only one where we found QOI resistance. So a lot of those fungicides that were controlling anthracnose are no longer effective. All right, let me move on to brown rot now. So that's all on anthracnose. As y'all are aware, the brown rot fungus is one that can develop resistance. It very readily does so. Where you have a concentration of a lot of trees like we do with a lot of replications of the fungus throughout the season, and where we use a lot of fungicides, you're going to get resistance development. Going back a long time ago now, but we determined that we had DMI resistance in collaboration with Guido Schnabel, and did quite a bit of surveying. And we did see in a lot of our production areas, we did have resistance. Guido also had a student work on this more recently, and what we determined out of that, without going into any details, but if you look at the blue or the orange bars, they're basically the same. It says we still have resistance, but it hasn't shifted that much. So our DMI resistance in Georgia is about the same as it was when we did our first surveys, going back now almost 20 years ago in some cases. But with that, we've also learned how to manage that resistance and to utilize these chemicals as effectively as we can. And so where we do have DMI resistance, we've observed that if you put out SDHI, QI combination products like Pristine, Maravon, or Luna Sensation, you can actually follow those with the DMI, even where you have DMI resistance, and you'll get pretty good control. That's hard to explain always, but there's something about it that does work, and it allows us to still utilize all 3 of these classes of chemistries. So in this first example here, these are just examples. This is 2005. You have an untreated control. You can see your 7-day after harvest amount of brown rot you have percentage-wise, 82.1. You look at Pristine followed by Propamax. You've got 30.8. And then you have just 2 applications of Pristine by itself. It's almost exactly the same, even though I use that DMI where I have DMI resistance. Here's another case in 2020, untreated control, Maravon by itself, or Maravon followed by Sevia, another DMI, and then you can see, again, the response is generally very much the same. So even where you do have resistance, you can still utilize a DMI if you use it in that way. So we've developed these recommendations over time, and if you look, especially if you come into the pre-harvest intervals, we'll say something like at 21 days before harvest, you may want to put out a Captan or a Bound or a Miravis type product, and then we've generally said Pristine, Maravon, or Luna Sensation at 14 days before harvest, and then maybe you can use a DMI during that time frame right before harvest. So that's kind of been the way we've approached this, and then either use Scholar or Chairman after harvest in order to control brown rot. Well, there's some new things that are coming into the market and some new things for us to look at. This is from 2021, and this is also from Byron. Here's our brown rot percentage after harvest and after taking it into these boxes and storing it for a while. You get 85.6%, and then we looked at Sevia and Indar in that particular trial, terrible control, showing that we have pretty strong resistance in that particular site still where we've done a lot of this work. Inspired Super also did not work well at all in that particular trial. So there's another product with a DMI in it. And then when you look at Maravon or Maravon followed by Sevia, again, you get not exactly the same results, but pretty dang close, not statistically different in any way. Just showing again that following with the DMI did work. And then we've got these other products, though, that these are the ones we're interested in. If you look at Luna Flex, this is an SDHI DMI combo, and you compare that to Luna Sensation or Maravon, you can see they're also not statistically different. So this Luna Flex is an example of again, the SDHI DMI combos are coming to market, and we're trying to figure out how do we utilize these without either increasing resistance or actually exacerbating the DMI resistance is already there. So there's the potential issues we have. And we got 3 of these at least that may be are coming to market, or some of them are already in the market. So objective here was to review these new SDHI DMI products, LunaFlex, Elysis, and Miravis Duo, and think about how could we possibly incorporate those. And I wanted to also look at some of the intrinsic activity of some of these DMI products in the field to see if they differed, and if we can incorporate any activity from that into our program. Now, all the trials that I do are generally going to be the same. They're randomized or randomized complete block designs. We put the fungicide treatments in there with an untreated check. We usually have 5 replications of each treatment, 1 to 4 trees, and then we leave unsprayed rows so we don't, and we use air blast sprayers. All of our equipment is basic standard farm procedure type equipment, and then we try to utilize cultural practices that y'all would use as farmers, and then we make our fungicide applications at 14 or 7 days prior to harvest, bring in 35 clean fruit in boxes, and then watch them rot, and that's how we collect our data. So looking at our 2024 flame prints trial, I'll make a note. We had 2 trials that were basically 2 sister trials, and one was in July prints, and we harvested twice, and we did not get brown rot. It was that dry during that period. We could not get brown rot. So if it's dry enough, you won't have brown rot. I don't care what you do. But we did have the later variety, and we got some brown rot. It's not an excessive amount of rot, especially for the trials the way we like to do them, but nonetheless, I think it's going to tell us something. So if you look at your untreated, when you go out here at 11 days after harvest, we had 37.7% brown rot. Okay, so if you look at, whoops, let me go back up here. If you look at Maravon, 3 applications did exactly what we thought it would do. It controls disease well. If you look at Sevia, we put out 3 applications of Sevia. It was statistically different, and it shows that we do have the resistance, but it actually did a little bit better than I thought it would. I actually thought it would be worse than it was. But then we have these 3 kind of a series of applications where we're trying to look at how to incorporate these products, looking at what we've been doing and trying to think through what we can do. So here we have Maravon, LunaFlex, and Sevia, or Maravon, Elysis, and Sevia in order as far as application order, and then Maravon, Miravis Duo, and Sevia in order. And what you get out of that is, if you look at this, they all look pretty good. And when you compare them to 3 applications of Maravon, with the exception of the one with the Miravis Duo, they're very similar. They're not statistically different. They don't come out and show that we're losing control or anything like that, although again, we didn't have a lot of brown rot by comparison to some of our trials. Here's LunaFlex. This is by itself 3 applications. Again, very similar to what we'd see with Maravon. So even though we know we have DMI resistance at that location, we're not losing control per se. If you look at the Sevia, here's your Sevia alone, and here's a Sevia plus Howler. And in either case where we had the Sevia, again, that was the worst material. Just the DMI by itself is not going to be sufficient by itself in that case. All right, so what are we going to do with that going forward? These products are going to be out there. We're getting questions already from producers on how do we use these things and should we use them. And I think in some cases they may be targeted at a price point that will make you want to use them by comparison to something like Maravon. Maravon is still the ultimate brown rot material that we have in the marketplace today, but also know there's some costs associated with it. So this is just what I'm suggesting now, just starting to try to think this through, and we may think this through in different ways. But going back to that original premise of how we would set up and line up these fungicides, perhaps you could use Captan, Abound, and Miravis here. Then maybe Pristine, Maravon, and Luna Sensation, so that's still SDHI QOI. And then now maybe Lunaflex, Alyssa's, and Maravus Duo. So you still have an SDHI, but you've got a DMI as well. So you're still trying to utilize as many of these classes as possible. The risk with this is we're putting out more SDHIs than we have in the past. And that could lead to SDHI development of resistance. So that is my concern. But as far as how I would do this, I think in order to mix it up as much as I can, this is what I would do. Going to another phase of the research we did last year, Guido had some work several years ago where he looked at the different activities of DMIs where we have resistant and non-resistant isolates of monolinea. And what he found in here, if you look at these bars, this is with curative applications on one, for example, where we did not have resistance, and then a couple from Georgia where we did have resistance. And what you would get out of that is, yes, there are some DMIs in particular that are going to be more effective. And I think in this case, I'm having a hard time reading my own graphics, but I think it's tebuconazole is the one that showed more activity in those trials. So this year, I just went back up in the Athens area, in Watkinsville, and looked at several. Oh, I'm sorry, I'm skipping over. This is another trial we done back in Athens years before, in 2012. And here I did look at particularly Maravon versus Elite, which is tebuconazole. You see it was almost identical. Now we don't, as far as I know, we don't have any resistance there, but it just shows that, yeah, some of these DMIs can be really very effective and can be comparable even to our best fungicide we have. And here we had good brown rot. I mean, 90.5% rot versus 1.6 and 7.9. That's high pressure. That's what we want to see. It's not what you want to see, but it's what we want to see. All right, so this last year, I looked at several of these DMIs, and I'll just try to tell you what we did here. This particular statistical analysis here, I did include the untreated. Here I did not, because it had so much variability and it really screwed up the analysis, but I'm giving you data from both. It's the same data with just a different presentation of the statistics. But what I get out of this is pretty clear to me, again, where we don't have resistance or not much resistance at all. If you look at the untreated, we had pretty good disease, 60.5% rot. Maravon took that down to 13%. But these DMI fungicides are then brought into different categories, and I think this is pretty clear. If you look at the Rally and Tilt, which a lot of people are still using propiconazole products, I think intrinsically they're just not as good, okay? If you're looking at something like Tilt, it may be cheaper, but you're not going to get out of it what you want as far as the amount of efficacy against brown rot. So that's the ones that are the worst. So Rally and Tilt were absolutely the worst, and they were statistically different than some of these others. If you look at kind of the second tier, Indar and Quash kind of rated together, and I think that's probably accurate. If you look at the materials that were the most active, Sevia and again Tebucon or tebuconazole, those came out best, and I think that is accurate. I think it's the intrinsic activity at these high rates, those are the highest rates we can achieve by label. I think those are the products that are going to have the best efficacy. Now, I'd also like to look at that more in the area where we have resistance to see if that holds up there as well, but we didn't do that this year. So our conclusions from that work are that these SDHI DMI products can be incorporated where resistance to DMIs is observed, but the long-term risk of resistance to the SDHIs may be increased. So you just have to realize that. Though the DMI products do vary in efficacy, we do not know whether we can take advantage of that where we have DMI resistance. But if I was growing peaches today, I would probably be looking at using either a Sevia or a tebuconazole type product if I was incorporating just a DMI into my rotation. But additional studies are needed, so I would say that. Okay, that's what I've got. So that's kind of an overview of our trials from last year on anthracnose and brown rot. Well, we did not look at Bravo independently. Bravo is generally going to be applied for the petal fall shuck. What's that? I'm sorry, monolinea infections in those particular trials. The answer is no. We did not, and I didn't see any brown rot. as far as those trials go, we had very limited brown rot. We did, in other words, we brought in the trays with healthy fruit, right? And so we could have been rating brown rot as well, but there was just very little brown rot. There was not enough brown rot in those trays to rate. We took numbers, but I think it's like occasionally we'd have like one fruit or something like that, but it just wasn't there. And it was very dry. I mean, our pre-harvest conditions on both of those trials for almost a month before harvest was just almost no rain at all, just hot and dry. Well, based on this, just what we observe here, I think it's a very long latent period. I mean, I think you can have early season infections, again, based on that would go season long. You just don't see any, and I think that's what I've learned. I'm always learning by observation. I never realized, and although I should have gone back and looked at Eldon's paper, and really, he says basically you don't see it until like at harvest. But what you're really saying with anthracnose is you do not see it in the field until the sugars come up and you're like right at harvest. I mean, in both cases where we took our data, this was like either the week before or during harvest, and we just didn't see this disease hardly at all. And yet we can achieve pretty high levels of anthracnose on what were apparently healthy fruit at harvest. But I think it's a very early infecting. I really think it can affect season long. But we just need, but I don't think it's always infecting at all times. I think it's just really when you have the appropriate temperature and humidity, rainfall conditions, you get infection. Similar to blueberries and strawberries. Any other questions? In the back. As far as any of the new products that are coming forward or our anthracnose? No, I mean, the only thing, we don't have anything new. We've always had Captan listed as something that would work. I guess what I'm saying here is it's probably, I think it's in the spray guide that we would put down that anthracnose would be. Oh, that kind of stuff. I'm trying to remember. We have put in the spray guide now some types of information on an at-a-glance spray schedule, right? Did we do that? So it should be in there. But I don't have this new information in there, and that's probably something we'll need to debate a little bit more. I'm throwing that out there right now from my perspective, trying to figure out how to do that. But we don't have it yet in the spray guide, and not all those products are registered yet. The Ulysses in particular, it may come to market next year or the year after. But it's registered on strawberry, but not on peach yet. All right, anything else? All right, I'm good.
Thank you very much. And up next, we have Dr. Juan Carlos Melgar is going to talk to us about bronzing and inking.
So yes, we're going to be talking about bronzing, inking. They're pretty much the same thing, two sides of the same coin, depending on the color that we see. This work has been done together with Guido, and then several graduate students that have been going through our labs over the last years. So I did a presentation a few years ago. Still, I mean, we keep learning about it. I'm going to review some of the things that we knew by then and some of the ideas that we were proposing at that time, and then see what's new. So for those that don't know, bronzing is this skin disorder that forms brown, bronze, yellow, irregularly shaped patches, typically around the equator in the central area of the peach. And even though it is something that we see at post-harvest, Guido with some of the initial works, and we're going to talk about it briefly now, we know that happens before harvest. It's in the tree before harvest, even though, again, it's really hard to see. The big question is what causes bronzing? And this is what I'm going to do, a quick summary of what we have learned over the last few years. First of all, Guido started working on this, and he couldn't find any pathogenic isolate in fruit with bronzing. So it is not pathogen related. As I was saying, while it's visible at post-harvest, it starts in the field. He took some pictures from the field in areas that looked something like suspicious. And then later, after going through hydrocooling, they were much more obvious. And so that was very eye-opening because, again, we know that it's on the tree before harvesting. We also know that it's not caused by air pollution or external contaminants, as the streaking, for example. We learned this when we had some trials with peach bags. And once we opened the peach bags at harvest, and we had some controls without bags, we saw bronzing at the same time and at the same rate. No matter if they were inside or outside, we had pretty much 50% of that crop with bronzing. So no differences between inside and outside. That means it is not coming from the outside. So we also knew that or learned that bronzing occurred even when we aggressively controlled thrips. So thrips was not, initially it was another thought that could be a reason or a cause, but it wasn't one of them. And then something that we have learned from growers, from our own experience, is that typically whenever there is rainfall shortly before harvest, it exacerbates the problem. Like sometimes we do the first peak, there is no bronzing. We do a second peak, there is no bronzing. 5 days later, it rains, we do a third peak, and then the third peak has a lot of bronzing. So why does it appear? And sometimes it disappears, like we have it on the first peak and then later we don't have it anymore. So that's intriguing, but again, we typically see that it's related to rainfall. Now, all that knowledge we learned all this over the last, maybe 6 years, something like that. A few years ago, that was 2019, we got 2 key findings. Guido was doing a spray trial with some coadjuvants to reduce the drop size of the size of the drop, to get a better coverage, etc. It was one of my orchards, and then we saw that one of the products that we were testing, this AG09075, which we didn't know what it was, because the company didn't tell us the name, just try this product. We measured bronzing there because it's a variety that typically shows bronzing, and definitely we saw bronzing there. And when we're looking at the incidence of bronzing in all these treatments, we saw that one had less. So we asked the company, what is this? And they told us that it was an anti-transpirant. So it was reducing transpiration, primarily transpiration from the fruit. We measure severity, and incidence is just if the symptom is there or if it's not, right? If it's there, we give them a 1 or 100, and if it's not, a 0. Now we also measure severity, and severity, it's how much. Like if there is no bronzing, it's a scale from 0 to 4. No bronzing is 0, up to 25% with bronzing would be 1, 25 to 50 would be 2, 50 to 75 would be 3, and more than 75% of the fruit surface would be a 4. So when we measure fruit severity, same thing, that treatment was working really well. We did it one more year in 2020, got the same results. So that was exciting. And that brought up some questions too, like some hypotheses. We thought the fruit transpires, and this transpiration, it's losing water throughout the fruit. If we have low relative humidity, there is more transpiration. If you have high relative humidity in the air, there is lower transpiration. So these, together with the fact that we see more bronzing when there is more rain, made us think that fruit-water relations could be playing a role in the development of bronzing. That was one of the hypotheses that we came up at that time. Another key finding was in a potassium trial that we had, where we were comparing, this was a nutritional study, and we were comparing a commercial standard rate of potassium with 5 times that rate of potassium to see what was going on. But again, we were not looking for bronzing, but we found there that trees that were irrigated and had that high concentration of potassium in the soil showed fruit with more bronzing than those that were irrigated with low potassium. And that's for bronzing incidence, and a similar thing we observed for severity. There were significant differences there. So together with the previous one, we thought, could bronzing be caused by this nutrient imbalance coupled with high transpirative demands? And I'm going to try to explain this with these drawings. If it rains, we get high saturation of the soil with a lot of water in the moisture in the soil. And then we have conditions of high transpiration, trees moving a lot of water. If we have this nutrient imbalance, maybe in the soil, we might be moving nutrients at a different, rate than when these conditions are not happening. So by taking a lot of samples and a lot of thousands of bronze peaches, and we decided to do a skin analysis, because bronzing is something that we see in the skin and not in the flesh. And from that skin, we analyze different nutrients, potassium, magnesium, calcium, and all these ratios among them. And again, I'm still summarizing, but I'm going to get there quickly. Over 2 years, in 2 different orchards, we did this in fruit that is like control fruit without bronzing. We collected the skin, like a few pieces of skin, free of bronzing. And then in peaches with bronzing, in areas without bronzing, we collected some skin, like for example there. And then also from areas that have bronzing, we collected those spots and ran a nutrient analysis. And out of all those nutrients and ratios that we saw a consistent, and I'm showing just here 2021 for scarlet prints, but we did it for June prints, for PF23, in 2022 as well, and always found the same thing, that magnesium and the ratio of magnesium by potassium was always higher in those spots. So, still we don't know if that's a consequence of bronzing or if it's a cause of bronzing. But there is something there related to nutrients, no? So now I bring here 3 questions. Can we do anything? And one of them is like, is that formulation that we were using over those 2 years efficacious enough to reduce transpiration and maybe reduce bronzing? How does relative humidity and the environmental conditions at harvest influence bronzing? And the third one, do we have any other proof, or can we find another proof that soil nutrition could be linked to bronzing? So that antitranspirant is a product called Transfix. We kept working on it in 2021 and 2022, and sadly we did not find any evidence that Transfix would reduce incidence or severity. Here is 2021, and you can see how, there are no significant differences for neither incidence or severity. We have about 90% incidence for peak 3. Peak 3 was a little bit higher. I want you to remember that for the next graphs. In 2021, peak 3 was the one that showed higher bronzing. In 2022, again, we did not see difference. It could have been because of the conditions in the previous years were different than in 2021 and 2022. But again, it is not consistent. We cannot recommend using Transfix or any other, so far, any other antitranspirant for reducing bronzing. In 2022, peak 2 was the one that showed the lowest values, about 50%, and also lowest severity, with peak 1 and peak 3 being the ones showing higher bronzing. Now let's look at the weather. Like, knowing that we had bronzing at that time, we went back to the weather station, get all the data, and see where we could find some explanation for this. Again, in 2021, peak 3 was the one with higher bronzing, and 2022, peak 1 and peak 3. So first of all, we look at rainfall these 2 years. So we have July 2021 and July 2022 here. And interestingly, you see how, again, peak 3, which happened at this, the harvest of, the third harvest happened at this time, that's July 16th. We had only, 10 millimeters, that's something like less than half an inch before that, but it happened, there was the same amount of rain for peak 1 and peak 2. So the big rain actually came after peak 3. You know? But then when we look at July 22, we pretty much didn't have any rain before peak 1, and we did have bronzing. And we had a long time of period, like 10 days without any rain before peak 3, and we did have bronzing there. However, for peak 2, there was some rainfall 4 days before, and was the time that we had less bronzing. So these data is telling us that, rainfall, it is not, it could be something related to rainfall, but not always when you have rainfall, we have bronzing, right? Looking at some of the parameters, temperature, and there are many lines here. This is like the average, the high, and the low of the day. We saw how it tends to show that there is high temperature at the times where we have higher bronzing. Like here in peak 2, at time of harvest, it was a bit lower, and these two are a bit higher. Here, not a lot, but something. But then when we look at the temperature minus the wet bulb, and this is a parameter that actually indicates, or could be, it's correlated to transpiration. You know, just because of you have high temperature, it's not going to transpire more. But when you put together both of them, the temperature minus wet bulb, the literature says that that's closely linked to the amount of transpiration. That one seemed to be a lot clearer, like, you know, at the peak here, we had a peak in bronzing. It happened here, and it happened here. So definitely these conditions of inducing high transpiration seem to be related to bronzing, more than just rainfall. So that's where the combination of factors come in. Now, related to the soil nutrition, we're doing some work with compost, and this is a soil health experiment. And interestingly, we have seen some reduction of bronzing in orchards with improved soil health, and this has been very interesting. Again, we use food waste compost that was implemented in the soil before planting, and then every year annually we applied as well. And then we have seen how, well, we have two rates of compost, compost 1, which is 5 tons an acre, compost 2, which is 10 tons an acre. And we have seen how consistently over the last 2 years, compost 2 showed less incidence here and here, no matter if you have little bronzing or more bronzing, because again, there is variability between years, but every year we have seen that reduction in incidence as well as in severity. Sometimes compost 1 does better than the control or comparable to compost 2 as well. But the trend is consistent in 2023 and 2024. That's for incidence, again, following the same scale as shown before. When we do severity, we also saw the same. There is between 10 to 15% drop in bronzing when we had the compost. So that means that maybe that balance, that more balanced nutrition, it's making a difference in the fruit. One of our goals this year is looking, for example, at that potassium, magnesium, and calcium, at the concentration at the skin. But in this orchard, like, is compost altering that concentration in the fruit skin or not? We expect so, and if we see that, I mean, we could link it to this to these findings. So as a summary, environmental conditions inducing high transpiration and probably transient nutrient imbalance in the fruit seems to, keep being one of the only clues that we can still link to bronzing over these 6-7 years. We have many times started with same hypothesis that after different tests it gets debunked. But these still, like all pieces of the puzzle, are still fitting for this first conclusion. And then that anti-transparent, or at least transfix, the one that we used, have been not consistent at reducing bronzing. Probably, again, it depends on the conditions and the timing. We typically do it 1 week and 1 day before harvest. But again, it may happen that we have, the environmental conditions were different over different years, and that's something that we need to, or we should look closely. And then that soil amendments have reduced bronzing incidence and severity. So again, even though we still don't have a full picture, we got a few little more clues to add to the puzzle. It is a nightmare, independently of if you are awake or sleeping. It is a nightmare that lasts for the month of July, and after that it's gone. And then, I always say, we need to go to Chile and continue studying there over the winter, because one more year, we just have to wait 1 more year till we get bronzing again. I want to thank the Department of Agriculture, Southern SER, for different grants that we got to study this, the Cherry Crossing Research Center for providing the compost, and then the Master Farm crew that had always helped us a lot with this. And with this, if you have any questions, I'd be happy to answer. Yes. So the question is if genetics could be playing a role in this. We have seen varieties that every year have bronzing and some that don't. So it is something that, there are definitely some that are more susceptible than others.
Genetics, because this is affecting the cuticle fertility, just for me.
We have seen it though in nectarines and in peaches, the same, and coming from different programs, from California to peaches from different origins. But again, there are some that show more than others. So it's some varieties. So yeah, we don't have those, but Guido in one of the trips he did to Spain, right? Or I don't know if you were in Germany or in Spain. But I remember you came back a summer saying I've seen bronzing in the supermarket there too. So
We saw in Kingston varieties, bronzing, by the legal heights that are very popular. We saw also these varieties.
So again, there must be something genetic when, at the same time, we have a variety trial, we're harvesting at the same time, and some are showing it and some are not, and they have all had the same environment. So there is. There is some genetics behind. Now we don't know if, I don't know, the trichomes. I don't know what could be affecting. We haven't done it yet. No. We haven't done a microscopic. Yeah, we haven't done a microscopic. We have been talking about it, but we haven't done it. Mm-hmm. Yes. Yeah, it is not something that. The point of that slide wasn't saying you should apply compost to reduce bronzing. That's another project, and it's just that since it was a variety, or it is a variety that we harvest in July, we check for bronzing, and we saw that compost has an effect in reducing bronzing, and it's a clue that's adding to the piece of information saying that a more balanced nutrition in the soil is affecting the fruit, you know. But I mean, my point wasn't that that's a strategy to reduce bronzing. But yeah.
Today, I mean, you've gone through and done a lot of work. Is there any take-home message that you can give
Harvest time, I mean, if you know that rain is coming, and again, not because of the rain, but because of right after the rain, you're going to have those conditions. And if you look at the weather, you see that the sun is coming out and you're going to have those conditions. If you can do a harvest, a day earlier, we still don't know if the harvest time influences the amount of bronzing. Like, I don't know, if we harvest early in the morning or late in the afternoon, the same day, are we going to have any impact? We haven't tested that, but if it's something that you can move there, that's probably the easiest thing that could be done. But as far as applying any product before, that's not working. Definitely, again, having a not over-fertilizing, having a balanced fertilization in the sense of not having an excessive potassium or applying potassium when you already have a lot. Or, I mean, we're looking into calcitic and dolomitic lime because of the magnesium that we have seen in the fruit. We think that there could be something there in the nutrition, but still don't have the full answer to be able to say that.
One last question. Do you think that during the transport, the contact that you switch, is more violent than another explanation? Or not?
It could be. We have not tested in the sense of let's harvest these roughly and these ones kind of like very, very carefully. It could be, but we have seen like when running the tests, like getting the peaches from the top of the basket or the bottom of the basket, and we see the same amount. We haven't seen a difference saying like, yeah, these ones that are on top and are not rubbing with each other as the ones that are at the bottom. We haven't seen any evident difference in between them. Same with, I mean, we were thinking like, could the sun after harvest, like sometimes you have to drive 10 miles from the field to the packing house. And those peaches could be exposed to the sun. Is that, I don't know, could there be some oxidation or something? We haven't seen a difference between the top and the bottom. And we have looked for bronzing in a lot of baskets. And so we don't have any evidence to say that. Yeah. So we have used Captan right before harvest to induce bronzing. Since we did not, at the master farm, for example, we do not have hydrocooling. We don't do things the same as at a packing house. We use Captan 10 days and 1 day before harvest, collect the fruit, put them in bins with water and ice, move them a little bit, and then put them in the cold chamber. Take them out and put them in the cold chamber. And we saw that way we always get some bronzing, pretty much. I mean, more or less, but it was a way for us to induce bronzing. And Captan definitely helps with that. And again, there we were seeing inking symptoms on the darker side of the blush, like the intense red, and then bronzing symptoms on the clearer side, on yellow-orange skin color. No? And so that's where we were seeing how it's the same, same symptom, no? It's just different color. So it is associated, you do not apply Captan right before harvest, but we did it just for inducing it. Was there any question with them? All right. Yeah.